Hello and welcome to this audio lecture on tissue biochemistry. This course is designed to give you a deep understanding of the biochemical makeup and functional mechanisms of various body tissues. In Module 1, we'll explore the biochemistry of muscles, kidneys, liver, and adipose tissues, highlighting their unique metabolic roles and interactions. By the end of this module, you should be able to describe the specific biochemical processes occurring in these tissues, their contributions to systemic physiology, and the consequences of their dysfunction. Let's begin with the biochemistry of muscles. One, biochemistry of muscles. Muscle tissue is essential for movement, posture, and heat production. The human body contains three types of muscle: skeletal, cardiac, and smooth muscle. Each type has unique biochemical characteristics, but they all share fundamental features related to energy metabolism and contractility. 1.1 Muscle structure and function. Skeletal muscles are composed of myofibrils, which contain the contractile proteins actin and myosin. These proteins interact through the sliding filament mechanism, powered by adenosine triphosphate, ATP. Muscle contraction is initiated by a nerve impulse that leads to the release of calcium ions, which in turn activate the interaction between actin and myosin. Cardiac muscle functions involuntarily and exhibits rhythmic contractions. It has a high density of mitochondria to support continuous ATP generation through aerobic respiration. Smooth muscle, found in internal organs such as the gastrointestinal tract and blood vessels, also contracts involuntarily and relies on slow, sustained contractions. 1.2 Energy metabolism in muscle. Muscle tissues use three main sources of energy. 1. Creatine phosphate system, for quick burst of energy. 2. Anaerobic glycolysis, breaks down glucose into pyruvate and then into lactate when oxygen is limited. 3. Aerobic respiration uses glucose, fatty acids, and amino acids in the presence of oxygen to produce ATP. In resting muscles, fatty acids are the primary fuel. During exercise, the muscle shifts to glycogenolysis, breaking down stored glycogen to meet energy demands. With increasing intensity, anaerobic glycolysis predominates, resulting in the accumulation of lactic acid. The Cori cycle plays an essential role here. Lactate produced by muscles is transported to the liver, where it is converted back to glucose and sent back to muscles. This maintains glucose homeostasis. 1.3 Muscle biochemistry in disease. Conditions such as muscular dystrophy, myasthenia gravis, and rhabdomyolysis involve disruptions in normal muscle biochemistry. In muscular dystrophy, defects in dystrophin compromise muscle integrity. In rhabdomyolysis, the breakdown of muscle tissue leads to the release of myoglobin, which can damage the kidneys. Two. Biochemistry of the kidney. The kidney plays a central role in filtration, excretion, electrolyte balance, and acid-base regulation. Biochemically, it is involved in urea formation, hormone synthesis, and reabsorption of essential nutrients. 2.1 Nephron function and filtration. Each kidney contains about 1 million nephrons, the functional units that filter blood. Filtration occurs in the glomerulus, where water and small molecules are filtered into the Bowman's capsule. Large molecules like proteins are typically retained in the bloodstream. This filtrate then passes through the proximal tubule, loop of Henle, distal tubule, and collecting duct, where various reabsorption and secretion processes modify its composition. 2.2 Metabolic activities in the kidney. Glucose reabsorption occurs mainly in the proximal tubule via sodium glucose transporters. Ammonogenesis. The kidney produces ammonia from glutamine to buffer urinary acid and maintain pH. Urea excretion. The liver converts ammonia into urea, which the kidney filters and excretes. Vitamin D activation. the kidney converts 25-hydroxyvitamin D into its active form, calcitriol, which is crucial for calcium metabolism. Erythropoietin production. The kidney releases this hormone to stimulate red blood cell production in response to hypoxia. 2.3 Renal biochemistry in disease. Kidney diseases like glomerulonephritis, chronic kidney disease, and renal tubular acidosis alter the normal biochemical functions. For instance, in renal failure, there is a buildup of uremic toxins, electrolyte imbalances, and metabolic acidosis. Proteinuria and elevated blood urea nitrogen, BUN, are common biochemical markers of renal dysfunction. Three. Biochemistry of the liver. The liver is a metabolic powerhouse involved in detoxification, synthesis, storage, and metabolism of nutrients. It plays critical roles in carbohydrate, lipid, protein, and hormonal metabolism. 3.1 Carbohydrate metabolism. The liver maintains glucose homeostasis through glycogenesis, synthesis of glycogen from glucose; glycogenolysis, breakdown of glycogen to glucose; gluconeogenesis, production of glucose from non-carbohydrate sources like amino acids and lactate; fructose and galactose metabolism, conversion into glucose or other intermediates. 3.2 Lipid metabolism. Key functions include beta oxidation of fatty acids, ketone body formation, especially during fasting, synthesis of cholesterol and phospholipids, lipoprotein formation, such as VLDL, HDL, and LDL. The liver is crucial in regulating plasma lipid levels and transporting lipids to various tissues. 3.3 Protein and amino acid metabolism. The liver synthesizes most plasma proteins, including albumin, clotting factors, and acute phase proteins. It also manages the urea cycle, converting toxic ammonia into excretable urea. 3.4 Detoxification. The liver contains enzymes such as cytochrome P450 oxidases that metabolize drugs and toxins. It also conjugates substances with glucuronic acid, sulfate, or glutathione to increase their solubility for excretion. 3.5 Bilirubin metabolism. Bilirubin, a byproduct of hemoglobin breakdown, is taken up by hepatocytes, conjugated with glucuronic acid, and secreted into bile. Impairment in this process leads to jaundice. 3.6 Liver function test. Enzymes like ALT, AST, ALP, and GGT are measured in blood to assess liver function. Elevated levels may indicate hepatitis, liver damage, or bile obstruction. Underscore 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 underscore. 4. Biochemistry of adipose tissue. Adipose tissue, often regarded as an energy store, is now recognized as a highly active endocrine organ involved in energy regulation, hormone production, and inflammation. 4.1 Types of adipose tissue. White adipose tissue, WAT, stores energy as triglycerides and releases fatty acids during fasting. Brown adipose tissue, but rich in mitochondria and involved in thermogenesis via uncoupling protein 1, UCP1. 4.2 Lipid metabolism. Adipocytes synthesize and store triglycerides during the fed state and break them down through lipolysis during fasting or stress. The process is regulated by insulin, stimulates lipogenesis. Glucagon and epinephrine, it stimulates lipolysis. 4.3 Adipokines and hormonal functions. Adipose tissue secretes hormones known as adipokines, such as leptin, regulates appetite and energy expenditure. Adiponectin, enhances insulin sensitivity and anti-inflammatory effects. Resistin, may be linked to insulin resistance. 4.4 Adipose tissue in disease. Excess adipose tissue, particularly visceral fat, contributes to obesity, insulin resistance, type 2 diabetes, cardiovascular disease, metabolic syndrome. Chronic inflammation in obese adipose tissue results in elevated pro-inflammatory cytokines, contributing to systemic metabolic disturbances. Underscore 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 underscore. Module 1 Summary. Let's summarize what we've covered in this first module. Muscle tissue relies on various biochemical pathways for energy, with distinct patterns for skeletal, cardiac, and smooth muscle. The kidneys play a crucial role in filtration, electrolyte balance, urea excretion, and hormone production. The liver is a central metabolic organ involved in nutrient processing, detoxification, and protein synthesis. Adipose tissue functions not only as an energy reservoir but also as a dynamic endocrine organ with significant implications in metabolic diseases. In Module 2, we will explore the biochemistry of the brain and neurons, reproductive tissues, and detoxification and excretion in tissues. We'll examine how these systems integrate biochemically and maintain physiological homeostasis.